What I want to talk about is, first of all, a little bit about building truly general theories of evolution, and then how we can make those applicable to the kind of special cases that we're all interested in actually pursuing. And first of all, because I may run out of time, and also because it's relevant to what I'll be doing, I just want to mention a few acknowledgements at the start. Two graduate students, Sarah Fumagalli and Ben Chin, contributed to this work. In particular, you'll be seeing some results from Ben's work later on. There will be data in this talk. That's not my fault. It was Ben who did that. But I benefit from it. So a lot of evolutionary theory, most modern evolutionary theory, is really a collection of special case models. And just as an example, when people are interested in studying neutral evolution by drift, especially in molecular evolution, they'll often use something like coalescent theory, which assumes no selection is happening, and it assumes finite population size, but it allows you to study drift, mutation, you can bring in migration, things like that. By contrast, when people want to study selection, they'll often use something like evolutionary game theory, where now you're assuming only selection, so you're ignoring drift, mutation, recombination, migration, and you're assuming effectively an infinite population size. So you have these different kinds of models, and they're built from the ground up under different assumptions. And a lot of times I've seen people argue that basically this is assumed to be an inevitable consequence of progress. As you get more and more sophisticated theories, of course, you're going to get more and more specialized cases that are not going to be compatible with one another. Now, I don't care for that approach particularly. I like the idea of everything being unified by general universal principles and being able to use those to derive special cases. So that's the way I'm going to be pursuing it. Now, first of all, to set up any kind of general theory, we have to start out with some sort of premises that we're going to start with. And I'll call these axioms, scientific axioms, because in a sense, these are things that we think are exactly true. They're not simplifying assumptions, which we know are not exactly true, but we hope they're pretty close. These are things that we think are exactly true. First of all, the things we're studying come in finite populations, right? The number of individuals is always some finite number. These things leave descendants, such that we can, in principle, talk about which descendants came from which ancestors. This doesn't mean one individual leaves one descendant. It could involve sexual reproduction or more complicated processes. But we can talk about ancestor-descendant relationships. Both ancestors and descendants have some phenotypes that can be quantified. So we can talk about the change of a consistent trait from generation to generation. And then the next two are different from what we've mainly been talking about. I'm assuming each individual has a probability distribution of fitness values, a probability distribution of possible number of descendants. So I'm treating fitness as a random variable. That is, it doesn't have a value prior to reproduction. It has a distribution of possible values. Similarly with offspring phenotype. Prior to reproduction, you can't say what the phenotype of a particular descendant is going to be. You might be able to say what the expected value is, but there's a range of possibilities. If we think about my firstborn grandchild, I don't have any grandchildren at the moment, whenever they come along, if they come along, they will grow up to have some height. I don't know what that'll be. I can make a guess about it. I might be able to make a pretty good estimate based on my genotype, my wife's genotype, what we know about our kids' growth trajectories, but that's just an estimate of the expected value. There's a distribution of possibilities, and I don't know what that's going to be, or what the actual value will be. So I'm treating fitness and offspring phenotype as random variables. And just to illustrate what I mean by that, in a case like this, in a population, an individual has an expected fitness. That's reasonable. We could talk about the expected fitness of an individual and the expectation of their offspring phenotype. And so a population would be like a cloud of points like this, where each point is an individual, and they have an expected offspring phenotype and an expected fitness. Making these things random variables, though, means if I take these words expected off, then I get something like that. Each individual now has a distribution of possible values. Those distributions can be different from one another, And those values can be correlated. In this case, they're negatively correlated, as you might get with offspring size, clutch size trade-off. A lot of organisms that provision the nest where the parents bring food, if they have a big clutch of offspring, each one's a bit smaller because each one gets less food. If they have a small clutch, each one's a bit bigger. So there can be different covariances and variances for these distributions for each individual. Okay? Now, doing this requires that we have a notation that keeps track of this difference between frequency and probability operations. And because I am going to use this distinction in an example later, I want to be clear about it. First of all, when I write phi, I mean a phenotype. And by itself, we'll assume it means parental phenotype. Phi superscript O is the offspring phenotype. And I'm using omega as relative fitness, W over W bar. Individual fitness divided by mean population fitness. That's the element of fitness that matters in evolution. And now we can talk about operations on these. If I put a bar over something, that means the frequency mean. I'll give examples as I go through of these. That's the mean across values in a population. Put a hat on it, that's a probability mean. That's the expected value of a random variable. And then we'll also deal with variances, thanks, and covariances. Thank you for moving that. Square double brackets are a frequency covariance. Angle double brackets are a probability covariance between random variables. And I'll show in a moment what that is. And then stacked values are regressions. And in fact, there are a bunch of mathematical relationships that I've derived relating these so you can work back and forth between frequency and probability operations. Putting them together and using those, we can derive a stochastic version of the price equation. And it looks basically like this. And just to use that to illustrate what these pieces are, this double square brackets, that's a frequency covariance. That term is the covariance between these points. Okay? That term, that first term, and this last one taken together are the entire deterministic price equation. So if you go back to Price's original paper, everything he had there is contained in that first term and this last one here. This term here wasn't there. This only appears when fitness and offspring phenotype are random variables, which I'll argue they are in nature, and we should treat them as such. So the square brackets are this frequency covariance. These angle brackets, that's a probability covariance. That's the covariance within these distributions. So in this case, for instance, it would be negative. There's a negative covariance between offspring phenotype and fitness for this individual. And there's a bar over this because this term is the average of these probability covariances. So just to give you an image of what we're talking about, and I'll remind you of this as we go along, because we're going to be looking in particular at terms like this that involve probability covariances. Now, this general equation is useful. This is, in fact, a generalization of the original price equation, which left out the stochastic component. But a lot of the questions we want to address in evolutionary biology don't naturally fit into this very well. For instance, when we think about selection, we think of that as a relationship between expected fitness, expected relative fitness, and phenotype. That doesn't appear anywhere in here. I've got relative fitness with offspring phenotype, but not with parent phenotype. In fact, parent phenotype doesn't appear in here at all. Similarly, inheritance, transmission, we think of as a relation between offspring and parents. Again, that's not in here anywhere, right? So if we want to start with an equation like this and then derive results that address the kinds of questions we're actually asking in evolutionary biology, we're going to have to do something. We're going to have to get parent phenotype in here somehow. The way to do that is that we seek to write offspring phenotype and relative fitness as functions of parental phenotype. And this is what people do in quantitative genetics and population genetics. And the simplest way to do it in the case of inheritance in quantitative genetics is to assume there's a linear relationship between offspring and parents. And if there's a linear relationship, then you can say offspring are parents times the heritability, the slope of that relationship, plus some random noise which they proceed to ignore. Okay? So as long as this relationship is linear, okay, that's a way to try to write offspring phenotype as a function of parent phenotype. Problem is, sometimes it's linear, sometimes it's not. Sometimes you get things like this that clearly have a nonlinear component to them. And it's not clear initially how to capture these in that sort of framework. Incidentally, the idea that all you have to look at is the linear relationship is so ingrained in evolutionary biology that I grabbed both of these from papers in the literature. In neither paper did they mention any nonlinear component, even in this one, to the data. They just drew a straight line through it and said, okay, there's the heritability, and we're done, and we'll move on. Because that's what quantitative genetics works with. It works with that linear component. But there's obviously something else going on here. So how do we capture that? Well, an obvious place to start, since heritability was a regression of offspring on parents, maybe we could do a regression of a nonlinear function, right? So we could do a simple polynomial regression, first, second, third order. That seems to fit. There's a problem here, though, and it's a serious one. If we look at this first order regression, it has a coefficient of phi, so that's the slope of that regression. Second order has a coefficient of phi squared and a coefficient of phi. Third order has a coefficient of phi cubed and phi squared and one of phi. Fine. The problem is this coefficient of phi, this first order coefficient, that should be heritability. That should be capturing what quantitative geneticists study as heritability. The thing is, you'll notice as I go from first order to second to third, that first order coefficient changes. It goes from 0.219 to 0.202 to minus 0.302. In other words, there's no clear biological meaning of that term. What I get for it is a function of what order polynomial I choose to fit. And since there's no natural order that all biological systems go with, I have no way to choose which is the best one. So in other words, this doesn't work because these coefficients don't have any clear biological meaning. Now, there's a way around that. And the problem, what causes the problem, is that different powers of something are not independent of one another. So if I just plot phi cubed versus phi, those are obviously correlated. Okay? One way around it is to construct a function of phi cubed that is independent of phi. That is, over some region, it's not correlated. That's what we mean by an orthogonal polynomial. This is a third order orthogonal polynomial, orthogonal to the first order. Orthogonal in this case means there's no correlation between them. And if we can do that, then we can use that third order one potentially to capture third order effects. Now, one thing you'll notice, though, is that was orthogonal over that region. If I pick a smaller region, then these are not orthogonal. In other words, orthogonality of functions is determined by the range of values over which you look. Strictly, it's determined by what's called a weight function, which I'm putting down here. And this is just shown here. The definition of orthogonality of functions is a function of this weight function. And so what weight function do we use? Well, there are different kinds of orthogonal polynomials. If you look them up online, you'll find a Wikipedia page and everything. And one set is the Legendre polynomials. They have a uniform weight function on an interval. These are used, by the way, sometimes in biology. Mark Kirkpatrick and some colleagues have used these in studying the evolution of reaction norms and things like that. Another kind is the Hermite polynomials, where the weight function is basically a normal distribution. But which one do we choose? Well, there's no off-the-shelf weight function that we can really make use of that captures what we want. The key, though, is we can make up our own. We can use the distribution of variation in the population as the weight function. And when we do that, the population is basically defining the set of polynomials. And when we do that, we can build zeroth order, first, second, third order polynomials. And these have a bunch of terms in here. What those are functions of the moments of the distribution of variation. And so this is a complicated function here, but I'm putting it in to show that you can write the third order polynomial completely in terms of the distribution of variation in the population, and that could keep going up and up. When I use orthogonal polynomials, it gets away from this problem that we had before. So now, if I project my data onto the first order, and then the first and the first, second, and third, the coefficient of the first order, again, is 0.219 here. And when I add a second order, the coefficient of the first order is still 0.219. And when I add the third, the coefficient of the first is still 0.219. So in other words, now when I add higher and higher orders, I'm not changing everything below it. So I can assign biological meaning to each of these coefficients. Okay? And in doing that, I can write offspring phenotype in terms of a set of orthogonal polynomials. Essentially, what I'm doing is constructing a space, a basis that is a coordinate system that is built by the distribution of variation in the population. And then I'm projecting offspring phenotype into that space. And I can project relative fitness into the same space. And I can put those together and get a general equation. This is equivalent, mathematically equivalent to that stochastic price equation I showed earlier. But now it's written in terms that make more biological sense, although it has some new terms that we haven't seen before. These, just to point out these here, these regression terms, regressions of expected or relative fitness on the orthogonal polynomials, those are capturing selection-like processes. There are actually some things in there that are not exactly what we call selection, but they're selection-like. They're involving the relationship between the fitness distribution and phenotype. These frequency covariances here between offspring phenotype and the parental orthogonal polynomials, those are capturing transmission, or what quantitative geneticists would call additive genetic variance. Strictly, the first order one is what quantitative geneticists would call the additive genetic variance. Now I put quotes around these because, first of all, this is not necessarily mechanistically additive, and notice there's not one of them. Right? Transmission is no longer a single parameter. There's a whole set of parameters capturing transmission, and most of them are not linear, are not additive. Secondly, genes don't necessarily come into here. Genes can contribute to this, but this transmission could be cultural, it could be environmental, or anything like that. So it doesn't have to be genetic. And finally, you notice these are covariances, not variances. So aside from not being additive, not necessarily being genetic, and not being variances, additive genetic variance is a perfectly good term. And because that's the traditional term in quantitative genetics, I will sometimes refer to it as the additive genetic variance term. But you can look at it and say it's not a variance, it's a covariance. The use of the word variance comes from the way Fisher originally derived this in a very special case where he could write it as a component of variance. In general, you can't. Broadly, this is capturing inheritance. So what we've now got is a description of evolution in terms of transmission terms and selection-like terms, which is good. There are also these terms. These are probability covariances between transmission and selection-like processes. And these are things that we've never seen before. The original price equation can be written in terms of that sum and that term. These are new. And in particular, these sort of terms don't show up, and we haven't talked about them before. I'm going to give an example of this later on. My main example will be looking at one of these terms and showing that these do affect evolution. One thing also, I'm not going to be taking apart transmission in this discussion. I'm just going to be saying it happens. But we can break it up. And so using the same mathematical formalism, if we have a developmental process, this is a phenotype landscape, writing phenotype as a function of some underlying factors for parents and offspring. And the offspring phenotype landscape does not have to be the same as the parent one. I made it a different color to make it different, but it could be a different shape as well. I was just being lazy when I drew the picture. We can write the covariance between offspring and parents, this transmission-like term, this additive genetic non-variance, non-additive, non-genetic non-variance. We can write that in terms of the development. These two terms are the development of parents and offspring, and the transmission of these underlying factors, be they genetic factors, be they cultural factors, environmental, whatever. So we can further break up inheritance like that. I'm not going to focus on it here. The one thing I will mention though now, because there's been a lot of discussion at this meeting of the importance of development in things like transmission. Sure enough, there's development playing an important role. This term, this relation between the underlying factors in offspring and parents, this is a function of standard transmission genetics. It's also a function of who you mate with, right? Because the distribution of alleles, say, or cultural traits in your offspring is not just a function of you, it's a function of you and your mate. And I just want to point out, mate choice plays as big a role as development in structuring transmission. And it's something that at this meeting, it hasn't gotten as much attention. It tends to get more attention sort of in behavioral ecology contexts, but we should be thinking of it in the same way. And I point out, it's for the same reason. Just like development is important in part because it translates genetic variation into phenotypic variation, mate choice is what takes the genotypic variation in one generation and builds a different distribution of genotypic variation in the next generation. And so don't discard it when we're talking about issues in transmission. But we want to extend this to multiple phenotypic traits. One difficulty in doing that is that there are a lot of ways to build orthogonal polynomials for multiple traits. All of these different pairs, the yellow pair, the blue pair, the red pair of bases, are each orthogonal, right? And in principle, I get the right answer if I use any one of them. And as you can see, they're orthogonal, meaning when I project these points into the blue, into the yellow, into the red, the points are uncorrelated. But there's no particular reason to choose one over the other. And this has stopped people from using multivariate orthogonal polynomials to study systems like this. There's a way around it. What we need to do is construct a pair of different bases. And I'm going to skip over most of the mathematics of this, but I'm happy to talk about it if you're interested, because it's what I spent a lot of time doing. I'm defining two different bases. A basis is a set of coordinates. A simple one, which is basically just the traits you're looking at. So if we're interested in height, weight, basal metabolic rate, then the simple basis would be height, weight, basal metabolic rate. They're not orthogonal to one another, but you construct a biorthogonal basis to that that involves height orthogonal to weight and metabolic rate. Metabolic rate orthogonal to height and weight, like that. That constitutes this conditional basis. And when we project one variable into one basis and the other into the other basis, then we get the right answer for calculating the covariance. These, by the way, are analogous to the covariant and contravariant bases that they use in general relativity. It's unfortunate that they use that nomenclature because covariant does not mean anything to do with covariance. But if you're familiar with that aspect of applied math, this is doing essentially the same thing. Using that, I can, and I'm skipping over the steps, but we can build the simple basis and the conditional basis. And this is just built in terms of the distribution of variation in the population. And then with that, we can project offspring phenotype into the simple basis and relative fitness into the conditional basis, and we can derive a general multivariate version of the stochastic price result, essentially, although it doesn't look at all like the price equation anymore. But this is now multivariate. It's following a bunch of different traits, and it's written in terms of these tensors that contain transmission-like terms and selection-like terms. And I'm just pointing out this series here, the first term in it, G2 with beta 1, that is Landy's equation for multivariate evolution of a bunch of traits. And this G2 is the G matrix we saw a reference to yesterday. So quantitative genetics results sort of come out of this as special cases. And in particular, the standard results in quantitative genetics is basically the first order term in that expansion. Now, as you can see, there are a lot more terms here. So if you have any system and you apply this to it, it blows up into a bunch of terms. But if you're interested in particular things about that system, you can often pick out which of those terms matter. So I want to illustrate how to do that. And the example I'm going to use is the idea of the evolution of division of labor. And this is an issue in evolution of cooperation. As you know, when people talk about the evolution of cooperation, they often talk about it in terms of a cooperative strategy or an altruistic strategy and a bunch of cheater strategies. And they ask the question, when can the cooperative or altruistic strategy increase when rare against these cheater strategies? Now, what we often see in complex social systems is not just one kind of cooperation. We see different strategies that are mutually cooperative. They work well together, and they wouldn't necessarily do well by themselves. This is characteristic of complex societies. It's also characteristic of complex multicellular organisms. And it's hard to think about how that evolves in the standard context of things like kin selection, because you have to understand, you're trying to understand how one of these traits could get fixed and then set the stage for the next one to get fixed. What I'm going to address is the possibility that two such traits can increase simultaneously with one another. And I'll show that that can happen, especially when we introduce stochasticity. So the question, how do such systems arise? So I'm going to look at a simple case. Consider organisms that can produce two compounds, both of which are necessary, A and B, but there's a trade-off. If you produce extra A, you have to produce less B, and vice versa. So there's a trade-off like this in producing. An individual by themselves produces enough A for themselves, enough B for themselves. Just by chance, because of mutation selection balance, some individuals are going to overproduce A, and some are going to overproduce B. Okay? In principle, if you're overproducing A, if you're next to somebody who's overproducing B, that's good, right? You benefit from their overproduction, they benefit from yours. But if you're not next to them, you're doing poorly. And when both of these start out, you don't expect them to be associated. So the question is, how could these two different strategies both increase? Now, in this case, there are actually four relevant phenotypes. Phi1 is the degree that you overproduce A. Phi2, the degree of overproduction of B. These are, of course, negatively correlated with one another. Phi-E1 is the amount of extra A in your environment. And Phi-E2 is the amount of extra B in your environment. So these are environmental variables. I am treating them as properties of an individual because they are. One nice thing about this sort of generalized construction of descriptions of evolution, a trait can be almost anything. If you can measure it, it's a trait. And if an individual has it, it's a property of that individual. And so I'm making these environmental factors traits that may be able to evolve themselves, and they certainly can alter the evolution of other traits. And then I'm going to assume that A and B are valuable in combination. So it's not helpful to have extra A if you don't have some extra B around. It's not helpful to have extra B if you don't have some extra A around. But if you have both, you have an advantage. Now, this is a classic sort of case that would be difficult to model using the standard sort of models that people use, because if you start out down here, there's no reason to move this direction or this direction. And because these are negatively correlated, you can't move this direction. So how can either of them get started? Well, let's write out the equations for them. There are four traits, four equations. 1, 2, E1, and E2. To simplify things, and because I want to zoom in on one part, if we assume that for the moment that these environmental traits are not heritable, they could be, and I'll come back to that, but if we assume they're not, and we note that these two are symmetrical, what I really need to focus on is this one. Now, all of these terms are going to be, in the way I've set this up, either 0 or negative. I'm assuming that there's no correlation between how much A you produce and, on average, how much B is produced by your neighbors. Obviously, if those are correlated, then they can get going. But I'm assuming we're starting out with no correlation. The question is, how can that evolve? Almost all these are negative, except for this last one. I'm going to focus on that. This is a probability covariance between this term, which, if offspring resemble their parents, is basically measuring how much the degree to which your overproduction of A is correlated with the amount of B around you. And this term, which is the selection, essentially, on how much B is around you. Okay? Now, I am assuming that on average, this one here, this term, is 0. Because we're assuming we're starting out with no covariation between these things. But this is a probability covariance. This is a random variable. So is this. And so they could have a probability covariance between them. And in fact, they will. And the way to see it, this is a simple case I made up, but just to be able to draw it, if we imagine individuals pair up and interact in pairs, and O means you're not overproducing A or B, you're just doing the original strategy. I could arrange them. It might be that they'll end up being arranged in such a way that I never have A and B together, in which case this covariance is negative and this regression is 0. It might be that you have just the exact number of combinations of A and B that you would expect by chance. In that case, this covariance is 0, and this regression is non-zero, but it's small. And then sometimes, just by chance, you'll get a bunch of A's and B's together, in which case this covariance is positive and this regression is larger. The reason the regression changes is the relation, the fitness relation, is nonlinear. And so as you move out on it, the regression slope gets larger. What this means is, in fact, this probability covariance is greater than 0. Just random variation in how individuals assemble with one another creates a situation where there's variance in this term and variance in this term, and when this one is big, this one tends to be big. So in fact, that term is positive, and it means there is a term in that equation that can cause both strategies to increase when they're rare. And by symmetry, the same thing applies to the other strategy. So there's a force that can cause two mutually beneficial but individually maladaptive strategies to both increase in frequency when rare, even if they're initially independent on average of one another. And this results, I should say, from stochastic variation in how individuals assemble themselves. So this would be invisible to a deterministic model. It's a case where you have to look at this sort of probability covariance in order to see it. And you see why I use this odd notation. It's important to distinguish frequency covariances from probability covariances. It's the probability covariance that makes this work. And so just saying stochastic selection at the level of individuals can lead to a case in which cells sometimes group together. And some of those groups contain multiple mutually co-adaptive strategies. That is a point at which you can easily see how you could get selection for multicellularity. Because now, if those groups ever reproduce by fission, there will be some heritability to the group phenotype. And then you've got selection at another level. Those last two equations that I put up there, those suddenly become non-zero because the environment now is heritable, and selection at this other level can kick in. So you can get going. And that can potentially facilitate at least one kind of multicellularity. Now here's the thing, I say one kind because you remember there are different ways you can be multicellular. And when we look across the metazoans, there are a couple of different kinds of multicellularity that show up repeatedly. One is aggregative multicellularity, where separate cells that can live independently often live independently for a while, then conditions are right or conditions are bad, they aggregate and form a multicellular unit, things like dictyostelium, what we're seeing in this case. But there's another way. There's also clonal multicellularity, where cells simply divide and they have a clone, a genetically identical cell that doesn't separate from them. It stays attached and then divides more. And you can build up a multicellular structure that way. The mechanism we just talked about makes sense in the context of aggregative multicellularity. It can get this process started. It's not going to get this process started because we assumed we were starting out with things that were complex and different enough that they're playing different strategies. Clonal multicellularity involves things starting out being genetically identical to one another. So this suggests that one mechanism for the evolution of aggregative multicellularity isn't really going to work for the evolution of clonal multicellularity. In fact, I think there are a lot of reasons to think these are really different things. This is just saying that this doesn't work so well for clonal multicellularity. These are really different things, and further confirmation of that, and this is where Ben Chin's work comes in, is when we plot these kinds of multicellularity on a phylogeny, they show up independently. So this is, and by the way, don't tweet this because this figure is not ready for prime time quite yet. Systemists will note some of those branches should be renamed. But what we did here was plot clonal in red and aggregative multicellularity on a tree of the eukaryotes. Key thing to mention first is they always appear in separate places. There is no place where clonal multicellularity arises within a lineage that does aggregative, or vice versa. So neither one leads to the other. They're really separate beasts. Furthermore, we looked at the properties of these internal branches in here, and green here is measuring the number of cell types on the branches leading to it. By the way, we're talking, most of these branches, of course, are unicellular. Yes, unicellular things can have multiple cell types. They only have one cell type at a time, but throughout their life cycle, they can have multiple types. And so we can look at whether either of these kinds of multicellularity arise primarily on branches that have a lot of cell types or few cell types. And we also looked at some other genomic properties. This is just showing some of the distribution of number of cell types for unicellular eukaryotes. What we see is clonal multicellularity, called divisional here, tends to arise in things that have fewer cell types than average. And although it's not really apparent here, aggregation is not statistically different from average. But clonal multicellularity arises in things that have fewer cell types, which is interesting. We see a similar pattern with the number of proteins or the number of coding genes. Clonality arises in things that have fewer than the average number of coding genes. So putting this together, I think it's worth saying these are really different paths to multicellularity. They occur in different kinds of organisms, and they occur for different reasons. Aggregative may start with a joint evolution of mutually beneficial traits that can increase together due to stochastic selection. By contrast, clonal probably starts with something like selection for increased size. Once it's there, though, it also sets the stage for multi-level selection to kick in and lead to differentiation. But that's going to come down the line in the case of clonal, rather relative to aggregative. So when we look at these, we really should treat them as different kinds of phenomena, different kinds of things. And if we try to further study what leads to their evolution, be aware that different processes may be involved. And so finally, I just want to point out the relation between the simplicity and the complexity of evolution. We started out with this simple relationship, the stochastic price equation. And many biologists kind of object to this because they say, well, there's a lot of complexity in biology that's not apparent there. Well, we can expand this in different directions. I can expand that term. I can expand that and that. Expanding it, it blows up into all these different terms. We've talked about one of them today. It's actually that one right there. This one is traditional selection. All these others are different biological processes that are going on in natural systems. So the complexity is there, but it all collapses back down into the same simple rule underlying it. I'm out of time, so I'll stop there. Thanks.